My name is Albert Porcar-Castell, and I am a professor of physiological forest ecology. I investigate how the light reflected or emitted by leaves can be used to study how plants, trees, and forests interact with their environment. Over the next minutes, I will briefly tell you why this is a relevant topic and how this emerging field of research can change the way we observe, investigate, and hopefully also manage plants and vegetation. Across history, forests have provided us with food, fibers, and energy. In addition, we now recognize that forests and terrestrial ecosystems provide us with essential ecological functions, contributing to the maintenance of the stability of our biosphere. For example, by capturing, via photosynthesis, about 1/3 of the carbon dioxide that our society is emitting to the atmosphere. But why am I reminding you of this multifunctionality? Because with a constantly increasing demand of forest products on the one hand, and with the challenge of climate change and biodiversity loss on the other, it is becoming more and more relevant to harmonize all of these functions and services, to ensure that our forests will continue providing us with a sustainable source of raw materials as well as essential ecological functions. But this process requires data. Data to help us understand the interactions and trade-offs between these services and functions, and data to support managers and decision makers. In other words, in addition to quantifying how much timber and biomass is accumulating in our forest, there is an increasing need to know also how those forests and their trees are doing. For example, how did our forest experience the lack of rain during last summer? At the moment, answers to these "how are you?" questions are obtained by using computer models capable of assimilating data both from ground and satellite observations. However, the level of uncertainty of these models remains high, in part due to limitations in the remote sensing data that we are still using, which mostly focuses on tracking changes in vegetation greenness. This is one of the issues that my research is trying to improve. But before I open up the underlying logic, let me share with you what is the long-term vision of our research community. Try to imagine for a moment that we would have a camera capable of recording photosynthesis and plant health status. We could point this camera down to a forest and record how different trees and plants are photosynthesizing over time. We could also mount this camera on a drone, a plane, or a satellite and record the spatial variation in photosynthesis across different forest stands, ecosystems, or the whole planet. That would be useful, wouldn't it? For example, this information could help us identify not only what trees inside a forest or what forest management practices are more productive, but also those that are less vulnerable to environmental stress, which could prove extremely valuable for decision-making towards climate change adaptation. How about if I now told you that these type of sensors are, in fact, already existing and operationally mounted on towers, drones, aircraft, and satellites? We call them hyperspectral systems and cameras. Hyperspectral because in comparison to the 3 spectral bands in your mobile phone camera, the red, green, and blue, these systems have hundreds of bands capable of detecting tiny changes in the coloration of vegetation. These technologies are constantly developing. However, the challenge remains as to how to quantitatively interpret this new data in terms of plant health and photosynthesis. This challenge is precisely at the core of my research. Next, I will briefly guide you through the underlying logic and introduce you what is so exciting about these new developments. We are all able to interpret the changing colors of plants. If the leaves of our apple tree have a deep green color, we conclude that our tree is in good health. The deep green color indicates that the leaf has sufficient chlorophyll, which is the pigment responsible for absorbing sunlight, powering photosynthesis. In contrast, if the leaves of our apple tree start to turn yellow, we conclude either that autumn is coming or that the tree is suffering from stress. Changes in the greenness of leaves and plants have been exploited by scientists for more than 50 years to quantitatively analyze the spatial and temporal distribution of vegetation and its photosynthetic activity at the local to global scales using remote sensing. The challenge with these methods is that, first, they are not optimal for evergreen vegetation, for example, a pine tree, which retains its green leaves and most of its chlorophyll even during the cold winter months when they are barely photosynthesizing. And secondly, chlorophyll contents, and therefore greenness, do not always respond to plant stress, which again limits the capacity of these methods to track photosynthesis and health of plants. But fortunately, leaves are not only green, and it is within these additional colors that we find a wealth of information related to plant health and photosynthesis, which now we can measure remotely. The first colors that we find underneath the green chlorophyll are those of the yellow, orange, and red carotenoid pigments. Carotenoids have multiple protective functions in the leaf, and their levels are rapidly adjusting in response to plant stress, making them an excellent indicator of plant health. Yet, although these pigments are always present in the leaf, we can only see them with our naked eyes during fall, once the contents of chlorophyll start to drop, revealing the beauty of carotenoids. Now, with the help of the hyperspectral systems that I mentioned earlier, we can track the dynamics of these carotenoid pigments at any time and scale, opening a new window of information to track plant health. But vegetation does not only reflect radiation, it also emits light, which brings us to the second of the hidden colors. Its name is chlorophyll fluorescence, and its color is red. Chlorophyll fluorescence is emitted by all green plants when under illumination, and the exciting thing about it is that its intensity, although small and not visible to the naked eye, is directly related to the regulation of photosynthesis, again opening new venues of opportunity to investigate photosynthesis and plant health at multiple scales. Also, as for the carotenoids, the remote estimation of chlorophyll fluorescence is now possible thanks to the new technological advances in hyperspectral systems and signal processing methods. So overall, and to conclude, next time you see a leaf, remember that there are other colors mixed within the green, the yellow, orange, and red of carotenoids, and the red fluorescence emission, which tell us how plants are doing. We can now measure these colors at multiple scales and use them to better understand how plants are coping with the environment, and by doing so, try to help them. As our understanding of these signals continues to improve, a new range of scientific and commercial applications unfolds, not only in carbon cycle studies, but also in spatial ecology, plant phenotyping and breeding, or precision agriculture and forestry. And I very much look forward to contributing to these developments. Thank you for your attention.